Professor Andrea Cavagna with us today, who is a research director at the Institute of Complex Systems in Rome, Italy. Professor Cavagna is, of course, a very well-known name in the theoretical physics and statistical field theory community. He did his PhD from the University of Rome, Sapienza, with Professor Giorgio Parisi in '98. And he subsequently did postdocs at Oxford University and at Manchester University. And he's currently, like I said, at this Institute of Complex Systems, and is also associated with the University of Rome, Sapienza. So Professor Cavagna's research interests initially were in non-equilibrium glassy dynamics, where he worked on free energy landscapes of glassy systems. But over the last decade, more than a decade, he has been working in physical biology, in particular with collective behavior in biological systems. So Professor Cavagna's group has been one of the first groups to sort of look at both experiments on bird flocks, on insect swarms, and to develop theoretical models of these interactions, this collective behavior in such interacting systems. In particular, he's interested in applying the concepts of renormalization group theory to the study of collective biological systems. Professor Cavagna has been awarded many awards and distinctions over the course of his career. In particular, I'll just mention one. So he was the recipient of the 2021 Max Delbruck Prize in Biological Physics by the American Physical Society. So today he's going to talk to us about these renormalization group concepts. So he'll tell us about natural swarms in 3.99 dimensions. So with that, Professor Cavagna, please.
Thank you. Thank you very much. It was the best introduction I ever had in my life. Sorry, but let me correct you. I didn't get many awards. I just got one. So that one that you mentioned. So I don't want to oversell myself here. Anyway, thank you very much also for this invitation, and congratulations for this initiative.
Thank
you. So, let me give you a little bit more intuitive description of what I will speak about today, which is collective behavior in biological systems. By the way, I wouldn't mind if you kept your cameras on, not because I want to check what are you doing during my talk, but because it's normally better to have a face feedback about what you're saying. And also I want to check that you're not reading the papers. Okay, so collective behavior in biological systems is something which go across many scales in both space and time. So this, for example, these are data for a colony of stem cells growing, and at some point you have a number of units of particle, which is quite large. You start having interaction, collective modes. This is something that goes on at a very small scales, but you have collective behavior at, this is a swarm of insects at larger scales, and this is what I will tell you about today. So the swarming phase. Of course, you have collective behavior in birds. So these are flocks of starlings in the sky of Rome, and part of our work has been done in birds, but today we'll focus on swarms. So collective behavior in this kind of system. So the question is, how do we tame such complexity? So what do we do in face of these enormously complex systems, where by we mean physicists, of course, because this is a question which is asked and answered differently in different communities. Well, typically what we do is that we do outrageously minimal models. So really things which are extremely more simple than the complex system we are describing. So for example, I don't want to give you the details now, but today I will end up writing the three differential equations for, let's say, the swarm phase, and for that I will mean, maybe all swarms of any species, just three equations, and this is something that we really need to justify in some way, because especially when we speak to the biology community, you have to convince people that, look, I mean, this doesn't make any sense. I mean, all this complexity, these differences in the biology and individuals, the collective level, and so on, and you just want to capture all this with three equations. So what is going on here? So from the physicist point of view, the idea, and this is something that we often write in our proposal and papers, the idea is that when you have many units interacting with each other. And when you have that correlations in the systems are strong, then microscopic details do not matter much. So this is the underlying assumption of doing very simple models, and it's the underlying assumption that most of physicists have in the back of their mind when they do that. The problem is, says who? So, okay, this may be an intuitive statement, but why? I mean, when you have strong correlation in a system, microscopic details should not matter much. Well, in statistical physics, in classical statistical physics, we know the answer of that. So, says who? Well, it's the renormalization group that says that. So we have a tool we have developed in the course of the last century, a path and the final tool, which is the renormalization group, which is exactly what proves that if you have strong correlations, you can actually have universality in the system. And it was a long, well, not so long actually, but it was a nearly a decade path happening in between the 60s and the 70s of the last century, and these are just a couple of milestone papers from Widom to Kadanoff, Halpring, Hohenberg, and then finally Wilson and Fisher. This gentleman developed first a phenomenological theory of scaling, and then all this effort had a pinnacle really in the development of the RG with this paper. And by the way, you can see from the title of that paper where I got the title of my talk. And so there was this, let's the red line. Correlation, okay, so some systems under certain circumstances are strongly correlated. You have a large correlation length. Scaling relations were initially introduced at the phenomenological level, okay? So scaling relations simplifying a lot the complexity of all your correlation function in terms just of few quantities like the correlation length, the relaxation time. And finally, there was the RG, the development of the RG, and the RG was actually the tool which, on one hand, proved why all this was working, and on the other hand, gave us a practical, a technical tool to compute things, and in particular to compute the critical exponents, which were characterized in these systems. And the biggest good and fruit of the renormalization group is the concept of universality. Within the RG, you really have a way to understand what is universality, and that is related to the, as we will see, to the idea of a fixed point of the RG, which attracts many systems which may seem different at the microscopic level, but they are not at the macroscopic level. So this red line is something something crucial in classical stat phys. The idea is, can we follow the same track in this kind of system, so in biological system? So not any biological system, but anyway, biological system where you must have many particles interacting and some correlation. So we have done, and other groups have done, experimental and analytical studies in the last decade, proving that actually the first 2 points in this path, so the first 2 steps in this path, so correlation and scaling, hold, at least in some systems, in flocks, in swarms, and under certain degree in bacterial aggregation, and so on. So yes, in some cases, actually quite often, in a large number of cases, these biological collective systems are strongly correlated. We prove that in the case of swarms, at least you do have scaling, so you have the validity of scaling relation. So the next step is, okay, given this premise, correlation and scaling, does the RG work? Can we use the RG to predict the critical exponent? And this is what I will talk about today. To do that, you have to start with a measurement, and in particular with some critical exponent. And so you have to do experiments. This is what we do in our lab, what we've been doing for the last 15 years or so now. So we go in the parks of Rome during the summer, taking photographs, stereoscopic photographs of swarms. We identify the bugs. These are, by the way, the bugs. These are small midges, non-biting midges of the family of chironomids. Okay, And once you do, you solve the biggest technical challenge, which is the tracking, the three-dimensional tracking of the individuals, you can transform the raw images that we take in the parks into a set of three-dimensional position and velocity of each particle or almost each particle in the swarm. So at the end of this, which I'm not giving you any details, but this part, let's say, can last for years, solving well, for us it lasted years, developing the tracking algorithm to do this. You have the data, the experimental data, and you have to decide what to do with that. So this is, for example, it's the data of a small swarm, right? Now reproduced in a new plot. But you see two swarms here because one of them is a real swarm. Okay, so it's the real coordinates of midges in 3 dimensions. The other one is just a bunch of random walkers in 3 dimensions confined by an harmonic potential. Because the first thing you have to prove is that there is some collective behavior. Because now imagine that all particles, all individuals or animals were just there flying around some marker, which, by the way, is true. It is well known that swarms form around some marker. It is a white cloth, a puddle of water, whatever. Or, for example, for mosquitoes, is a lamp, right? So you have many mosquitoes running around the lamp. So you know that there is a marker. Maybe they're just each one individually flying around the marker without interacting at all with the other individuals. So this is our zeroth model. So a batch of random walkers in an harmonic potential, no interaction between the particle. So in the Hamiltonian, you just have sum over I, not Ij, right? And you see, one of those here is that system, non-interacting system. The other one is, and I'm not telling who is who right now, but the other one is a real bunch of swarms. So clearly you see that by eye is not entirely straightforward to see which is the non-interacting case and which is the real biological case. And you can actually understand a little bit from the heterogeneities maybe, but anyway, the left one is the random, is the bunch of random walkers non-interacting. The right one is the real swarm. But clearly it's not that collective behavior is something that in the swarm case you clearly see by the fact that there is some synchronization, like in flocks. In flocks, they all do the same thing. They all go in the same direction. Here is not like that. So what you need to check whether there is collective behavior is to calculate the correlation. And if you calculate the spatial correlation function, so you compute the velocity of each particle and you compute the velocity fluctuation, so the fluctuation with respect to the mean, you can plot a correlation function as a function of the distance between the particles. And then here at the level of this spot, you clearly see the difference between the natural real swarm, the black points, and the non-interacting batch of random walkers, where the correlation function, of course, apart from some noise because of the statistics, because this is a finite size system, but it's zero, as it should be, because there is no interaction at all. So this is clearly telling you, first of all, that before saying that there is no collective behavior in your system, well, you better check the correlations because it's not that because you have zero order parameter, there is no collective behavior. I mean, as physicists, we know that, right? In fact, we know that the most correlation in the system is of a critical system is around the critical point where you still don't have spontaneous magnetization. So we know that the order parameter is not the most profound hallmark of collective behavior. And indeed the correlation is. So we have non-trivial correlations. By the way, correlation that extends quite beyond the nearest neighbor distance, so the interparticle distance. In this case, we know that the interaction is acoustic and is very short range for midges. They basically, hear the wing beat of the other individual. So this is the kind of interaction they have here. So the correlation extends quite beyond the nearest neighbor distance. And we do find that in all our swarms. And in all our swarms, yes.
Can I ask one question? So what's keeping the Brownian particles, like random particles, confined?
In our example here, a harmonic potential. a parabola, so a harmonic potential. This is done with an harmonic potential.
So all the particles are in a single harmonic potential?
Yes, single harmonic potential with the same center, right? So they are all really oscillating, not oscillating, but walking randomly around the same origin.
Okay.
Okay, well, for the midgets, we don't know, of course, but we presume that they also interact with the landmark, but they also interact with each other. Otherwise, there wouldn't be any correlation. So we find non-trivial correlation in all our swarms. If you compute the integral, which is a measure of how much correlation you have, and you take the maximum of the integral function, you can plot that in all our data, in all our swarms. So each point is a single individual swarm. And you see that in all cases, you clearly have a value of the correlation, the total correlation, which is significantly larger than the random case. The integral of that in standard statistical physics would be the susceptibility. Here, it's probably an abuse of language because there is no fluctuation dissipation theorem telling us that that is the actual susceptibility. We call it susceptibility in our papers anyway, but it's just the integral of the space correlation function. But there is more. Even though there is a significant noise, what we find is that the correlation length seems to be scaling with the size of the system. So it's not simply quite large, but is scale-free, the correlation. So we don't have just one scale of correlation length, which is the same in all our swarms, because we have swarms of different sides. We can see how that scales with the sides, and we find that this is a scale-free system. Notice that, unlike in birds, this scale-free behavior, it may not be entirely trivial, because in birds, you are clearly in the order phase, in the order phase of a continuous symmetry which is broken, because birds are choosing to fly in one direction. So if you are in standard stat phys or also in particle physics, you are breaking a continuous symmetry. You know there must be Goldstone modes, and Goldstone modes meaning massless particles. In stat phys means an infinite correlation length, so a scale-free system. But here clearly you are not in the order phase. So this scaling with L means that you are kind of near critical in some sense. But important point is that we have scaling of the correlation length.
So what is, sorry, if I may, what is the L over here?
Is the size of the swarm. It's the linear size of the swarm.
So it's not the number of whatever.
No, it's not the number of individuals. It's the linear
Okay.
size of the swarm. It's the linear size of the swarm.
Okay.
So the next step is to say, okay, we have a significant correlation in space. What about correlation in time? So you can now compute also the space-time correlation function, which is fluctuation at this individual in this position at this time, how correlated they are with the fluctuation of another individual at a certain distance at a different time. From this, you can extract a relaxation time, tau, and you can plot the relaxation time of a given swarm as a function of the correlation length of that swarm. And what you see is that space and temporal relaxation are entangled with each other. And this is something that we know also to occur in standard statistical physics. That tau normally scales like psi to the z in a near critical system, which is called critical slowing down in stat phys, and which is a very deep law. It's telling you that you cannot have two independent relaxation for space and time. And the intuitive justification is quite easy, in the sense that if you have to relax larger regions, so with larger psi, you need more time. But the important point is that there is an exponent connecting the relaxation time with the correlation length, which is one of the crucial exponents in the theory of critical phenomena, which is called Z, the dynamical critical exponent. And this is the one that we want to calculate then theoretically, okay? And notice that it's also one of the most approachable from the experimental point of view for us, because you calculate the relaxation time, the correlation length, and you have Z. For example, if you wanted to compute gamma, we will have to compute the susceptibility, but then you have a problem of how to define the susceptibility in the swarm, while Z is quite easy to approach experimentally. Fitting Z is not entirely a piece of cake because if you just do, so you plot log of tau versus log of psi and you have to make a linear fit. So this is the basic procedure in which you fit critical exponents. If you don't have the experimental errors and you just do least square, okay, that seems a very reasonable thing to do. But the problem of least square is that it's assume homogeneous error in X and Y. And basically, it gives different exponent if you fit X of Y or Y of X, which is not very nice, because the problem of least square of LS is that it assumes that all this, the variance, all the scatter is in the Y axis, which in our case is not absolutely not reasonable, because we certainly have an experimental error both in tau and in psi. And indeed, you see that if you plot tau of psi, you get this critical exponent about 1.16, which is what we did in previous papers, by the way. Okay, we didn't thought about that. We say, okay, yeah, you want tau of psi, let's do this fit. If you fit psi of tau, you get quite a different exponent, 1.62. And again, this is because in one case you're assuming that all the scatter is in tau, in the other case you're assuming that all the scatter is in psi. So the right thing to do then is to calculate an experimental error on tau xi, long story, but it's not at all trivial experimentally. But once you do that, you can use smarter methods like effective variance, in which now you do have about the same exponent when you fit either of the axes, and this exponent is 1.32. So the next step is to have an error bar of this exponent. To do that, we do a resampling method. In other words, we have n data, and we take subgroups of n-half data, and we fit the exponent in each subgroup, and then you have a distribution of exponents, right? And the variance of the standard deviation of that distribution gives you a hint of, well, gives you the error bar of the exponent. So here I'm reporting the two cases, which effective variance, which is what we believe is the best way to fit the exponent now, and the estimated we have with least square. You see that the error bars, even on the exponents, are very significant, okay, but it is what it is. On the other hand, you can see the data. You have outliers, we have large error bars. So remember, each point here is the data from one individual swarm in a real park in Rome one afternoon over the year. So it's not, these are not numerical simulation, okay? Fine, so experimental summary, we have this kind of law connecting temporal relaxation and spatial relaxation with a critical exponent, which experimental is 1.32 plus minus 0.18. Theory. So what we want to do is to compute this bugger here and to find out whether we have a theory which reproduces that. So I will be as sketchy as possible here because it's a very long story, but let's say that there are three big ingredients in the theory we're developing. The first ingredient, which is the easiest, is ferromagnetism, which at the biological level means just imitation. So the idea is to write something which is just a ferromagnetic interaction. So each individual try to align its direction of motion to the one of its neighbors. Okay, and this is exactly the mechanism behind all ferromagnetic models like classical Heisenberg and so on. So the dynamics then would be that the time derivative of the pointers, so the direction of motion, which can be fixed to 1, okay, for the time being, is just an average of your nearest neighbors, where Nij now is your interacting network. So Nij is 1 if you're interacting with your neighbors, is 0 if you're not interacting, plus some noise. So this is a very simple equation, which is basically the standard ferromagnet. Okay, simple equation, but not simple model to solve. We know that this is, I mean, for discrete variable, this is the Ising model, and this is very hard because Nij is a short-range interaction matrix, right? It's not fully connected, okay? However, this kind of model, you can do RG, so you have a flow of the coupling constant. This was the very first RG calculation, right? The Ising model done in '72, and then Halpering and Hohenberg and Matt did the same calculation at the dynamic level on the same year. And at one loop, which is the approximation I'm treating to you today, what you find here is that Z is equal to 2. Now, this number 2 is actually just dimensional analysis here. And you can see that because on the left-hand side, you have a time derivative, one time derivative. On the right-hand side, you have something which looks complicated, but is basically just a discrete Laplacian. If you can rewrite the interacting part, the short-range interacting part on the right as a discrete Laplacian. And you know that at the continuous level, this equation is simply S dot equal Laplacian of S, of sigma. And so you have first derivative in time, second derivative in space. So clearly the exponent connecting dimensionally space and time is 2. So this 2 here comes from the fact that RG at one loop here gives no perturbative correction. Anyway, apart from this, what you have is that by using simple ferromagnetism, you have a critical exponent equal to 2, which is quite far from the experimental determination that we did. So something is clearly missing. So what is missing, obviously, is activity. So the model I just described to you was a standard ferromagnet in each pointer, each arrow was just sitting on a fixed lattice, which is clearly not the case, right? Because these are active systems. So it means that if your bug, let's call it Luke, at some time is interacting with these 5 other midges, okay, and one of them, let's call it OB1, but they are all flying in some direction, okay, so everyone is moving. So at the next time step, you may have that OB1 gets out of the interaction range, while Yoda, which was out before, gets in the interaction range. So you see, even at fixed density, so even without assuming large density fluctuation, you do have a rewiring of the interaction network. You may also have a density fluctuation, of course, and complicated phenomena like phase separation and so on. But the important point is that even in the simplest case of homogeneous density, which is the one I will treat later, you do have a rewiring of the network. So how this is relevant for swarms? Well, you get an intuitive idea that it is because they're going all over the places. But actually what we did was to compute the time scale of relaxation of the velocity and to compare it to the time scale of relaxation of the network, of the interaction network. And we did that over different spatial scales. Because, for example, for flocks, you would say that also flocks are a very active system. But in flocks, because they're all going in the same direction, you can actually prove from the data that the time scale of reshuffling of the network is much longer than the time scale of relaxation of the velocity. So this is why in flocks, a pseudo-equilibrium treatment makes sense, while here is not at all like that. You can see that on every scale, the time scale over which the network of interaction reshuffles is about the same as the time scale over which you have the relaxation you want to describe. So clearly activity is very important in this system. So you have to put activity in, and how you do that? Well, this was the Vic-Check model. This was the very first model introduced by physicists to describe active matter. So you say, okay, my interaction network now depends on time. And how it depends on time? We have exactly the same equation of alignment with your neighbors, but now you say that the position of each particle actually follow the arrows with some modulus V naught. So now V naught sigma I is your velocity. So now you move from just pointers fixed on a lattice to actual particles going around. And this is the Vic-Check model, which if you want is just like a ferromagnets on the move, because the interaction network is changing in time. To do RG calculation, you have to go from the microscopic model to the field theory, to the mesoscopic model. Like you have to go from the Ising model to lambda phi 4, to Landau-Ginzburg Hamiltonian. And this is what Toner and Tu did after the Vic-Check model was formulated. So you can rewrite the Vic-Check model in this way. This part here, the force, if you want, you can write it as the derivative of some Hamiltonian with respect to the velocity, where the Hamiltonian you see is just, again, a ferromagnetic Hamiltonian. Every VI try to be as close as possible to the VJ of the interacting neighbors. So what Toner and Tu did was to transform this microscopic model into a field theory, a dynamical field theory, mesoscopic model, by doing some phenomenological coarse graining. that is not an ab initio calculation. It's down a la Landau, let's say, okay, saying, okay, by symmetry, there will be this term, these other terms, and so on. So it's not a proof. But basically what you do is that you transform the normal time derivative into a material derivative, as you would do in fluid dynamics to obtain Navier-Stokes, because this is basically a Navier-Stokes plus a Landau-Ginzburg theory. So you have the material derivative, which takes into account that you have the normal time derivative, but also transport. You are transported by the velocity field. You have a coarse-grained Hamiltonian, okay, which is the standard Landau-Ginzburg Hamiltonian. So you do have a more or less fixed model of the velocity. And of course, you must also have an equation for the density. So the original Toner-Latour equation had two fields, the velocity fields and the density fields. And of course, this is why they are so difficult to solve and to study with the RG. So today we will just study this equation in the very simple approximation of incompressible case. So in that case, you assume that the divergence of V is 0, so you have that the density is constant. Is an approximation, of course. Is it meaningful? Well, we do not find in our data for midges evidence of large density fluctuation. Certainly there are density fluctuation, but not, we don't find any evidence of phase separation, MIPS or things like that. So this is the best we can do here, and it makes sense. And what Chen, Toner, and Lee did in 2015 was to, under this approximation, to study the new fixed point, the new RG flow. And they found that the Z equal to fixed point, the old one, was unstable against activity. And they found a new fixed point with critical exponent 1.73. So how good is that? So you see, we are moving in the right direction because we're going down in with exponent, but we're still quite far from the experimental determination. Even being generous with the error bars, this is very much not compatible with the experiments. So something is still missing. What is that? And this is our last and third and last ingredient, which is inertia. So what do I mean by that? Well, let me first give you a very intuitive sketch of what I mean by that. So this interaction in these systems, you can imagine them as a transfer of information from one individual to another individual. So a sort of domino effect, okay? And when you have this transfer of information, you always have a competition between friction and inertia. So this is the case of the real domino, okay? If you have a domino without friction, of very low friction compared to inertia, you have a very efficient transfer of information. But if you do that underwater, in which you have high friction compared to inertia, you do not have an efficient transport of information. So the transport of information, which is also how the fluctuation propagates statistically in your system, is normally heavily affected by the competition between friction and inertia. Why we expect that to have an impact on the critical exponent? Well, remember, the critical exponents connects relaxation time and correlation length, so time and space. Now, this is not a dispersion relation, of course, it's a critical relation. However, forget about that for a minute and just take the dimensional gist of this equation. At the dimensional analysis level, we're saying that R, space, scales like time to the 1 over Z. So you can imagine that every mechanism which enhances the propagation in time of fluctuation, right, will make grow this exponent 1 over Z. So in other words, will decrease Z. So again, this is not a proof because this is not a dispersion relation, but it is actually something that at the qualitative level works. So in all the zoology of critical, dynamical critical phenomena studied in the 60s and in the 70s, you can clearly see that whenever in a theory you added an ingredient which enhanced the propagation of the fluctuation, you always were lowering the critical exponent Z, which is exactly what we need here. However, the question is, do we have any experimental evidence that inertia is relevant in this kind of system? Because you may say, okay, fine, you see that it would be nice to have inertia to lower the value of Z, but that is not a good enough justification. So do we see any evidence of inertia at the experimental level? Well, to check that, we have to investigate better the correlation function. So let me give you a very simple case. The stochastic harmonic oscillator, just one particle. So the easiest stochastic differential equation ever, you have inertia, Q double dot, viscosity, Q dot, and stiffness, right, KQ. So this is just the spring keeping you attached to the wall, equal noise. So this you can solve exactly, of course, and if you compute the time correlation function, of course you have different regimes depending on how strong is your friction compared to inertia. So you may be in the deeply underdamped case, in which clearly the relaxation is non-exponential because you have oscillation due to inertia. Or if you now boost viscosity compared to inertia, you can go to the quasi-exponential relaxation, in which you do not see any sign of inertia apart from the very, very, very short time behavior. This may be something hard to detect experimentally if you only look at the behavior of the correlation function, but if you do a trick, it becomes very easy to detect. And the trick is just, well, define first of all the scaling variable, which is time over relaxation time, because all this will be a function of t over tau. Then take the log of the correlation function and divide by x. Now, if this is an exponential, e to the minus x, you take the log of minus x, you divide by x, takes the minus, you get 1. So this quantity h, which I'm defining, is 1 for pure exponential relaxation. And indeed, you see that in a very overdamped case, you have something which is equal to 1 down to the very, very short times. However, if you are in the underdamped regime, so inertia is large compared to friction, so viscosity, clearly this is not equal to 1, because this would be an x squared, you divide it by x, and it still goes to 0 when x goes to 0. So this h is a very, very quick way to see whether you have inertia. What the data tell us? Yes, you do have a clearly inertial relaxation in natural swarms, which is not compatible to the Vic-Check model, okay? And so what we have to do is to reinstate inertia in the theory. How do we do that? Well, we do that as we would do in classical mechanics. So an overdamped equation, the classical Langevin overdamped equation, is this one, Q dot equal force plus noise. So in the overdamped case, the force acts directly on the coordinate. But we know that the original Hamiltonian equation were not that. In the original equation, you had that Q dot is equal to the momentum P. And then P dot is equal to the force. And if you want to thermalize the system, you have friction and noise. And friction and noise may or may not be related by some fluctuation dissipation theorem. But the important point is that this is the full equation. And when friction becomes very large, then you go to the overdamped case. So we want to go back from this to that. And this is what we did. We introduced a new model, which was equivalent to reinstating inertia in the Vic-Check model. And the idea is that in the Vic-Check model, you have that, the force, the alignment force, directly acts on the velocity. While here what we say is that we have a momentum, the generator of the rotations, because here the symmetry is the rotational symmetry. So rotational symmetry means of each individual direction of motion. I'm not talking about rotation in the outer space around the origin, but rotation in the inner space of the order parameter. So we are introducing a new variable, which we call the spin, which is actually the generator of the rotation in the internal space of the order parameter. And now we say that the spin is the one which rotates the velocity, and now the force acts on the spin, exactly as we would do in the Hamiltonian case. But then we have some relaxational terms acting on the spin, so that when the friction eta goes to infinity or becomes very large, we go back to the Vic-Check model. So S is really the momentum associated to the rotational invariance of the system. If we do this, we have a new model, and you can clearly see that the relaxation of the new model is much more similar to the experimental relaxation than the original Vic-Check model, especially if you compute that quantity h I told you about before. So Vic-Check model has a totally exponential relaxation while the data have a non-exponential relaxation, a very inertial one, the new model reproduces that. So then we do have also experimental evidence that inertia is important in this system. So we have this new model that we call the inertial spin model, in which, again, velocity is turned by the spin. And now if you introduce this Hamiltonian, which is the old Hamiltonian of the velocity, the alignment part, the ferromagnetic part, plus now a kinetic term, that would be equivalent to the p square over 2m term, okay? Is the momentum square. Then you can replace these terms here in this way. And so you start to see some symmetry in this equation, which is a classical Hamiltonian symmetry. You have the dv over dt is dh over ds, the cross derivative, the reversible terms. ds over dt is dh over dv. And then you put also some non-irreversible relaxational terms over the spin, so you can go back to the Wicksec model whenever you want, okay? But we believe that we are not in the overdamped regime, but we are in the underdamped regime. By the way, if you do this kind of model for flux, you have a big difference. So this is what happens in a simulation of the Wicksec model when you try to turn one bird. So you see, you fail to push the whole flock following you because the system is overdamped. If you now run our model, which was developed, I told you the justification for swarms, but we developed it even for flocks, okay? If you do the same things for our model, you see that by turning just one bird, you succeed in making the whole flock turning. It's not like in the domino effect. In one case, you're unable to propagate the information. In the other case, you smoothly propagate the information through the system. But let's go back to swarms. So what is the effect? What are the effects of inertial dynamics on the swarm case? So again, we have to go to a field theory to do RG. So we do the coarse graining of our new model. So we will have two fields now, the velocity field and the spin field, the generator of the symmetry. We will have some reversible terms and some irreversible relaxational terms. And this is the Hamiltonian, in which you have the Landau-Ginzburg Hamiltonian as before, as in the Toner and 2 case, plus some kinetic case. Here, I'm still at equilibrium because these are normal derivatives. And the reason why I'm also using the stepping stone is that the model on the right is an equilibrium model and which is well known. It's called Model G in the zoology of critical phenomena. And it was described by Halperin and Honenberg in the famous review of 77. And this is a model, this is a class of model for describing superfluid helium and quantum antiferromagnets. So it seems complicated, and it is, but we're moving in a very well-known class of equilibrium models in which you cannot describe the behavior of a system by just one field. Because you have a conservation law, you need another field. And this is exactly what happens in superfluid helium. And this changes the dynamical critical exponent from 2 to 1.5, which is the effect I was telling you before. So by introducing a conservation law and so facilitating the propagation of information, you decrease the critical exponent. But the last step we have to do then is to add activity, so the off equilibrium part. And we do that again, like Toner and 2, by substituting the standard time derivative with a material derivative, in which now you have transport. But you have transport both of the velocity and the spin. So the moving field, so this field of particles, in transporting velocity itself, but also the generator of the local symmetry, which is the spin. And so we end up with this coupled stochastic differential equation. And again, remember, we are working at incompressibility. That is, density is constant, so we don't have 3 fields, but only 2 fields, which is bad enough, I can tell you. So by doing the calculation, by also adding the density field would be impossible, at least now.
I had one question. Can I?
Yes.
Yeah, so can you explain once more that gamma V, why did you bring that, gamma V and gamma S?
Okay, so it's, let me move this. Okay, so this is if gamma was equal to 1, this would be the standard definition of the material derivative.
Yeah.
In which the total time derivative will be the partial derivative by v dot divergence. Okay.
Yeah.
Now, now do you have, so in Navier-Stokes you would have just that. However, first of all, and this was already noticed by Toner and Tu, you don't have Galilean invariance in this case because the system is active. They have a reference frame, which is, you know, air or whatever you're doing the experiment, but you do not expect to have Galilean invariance. And so you're not allowed to leave a 1 here. There may be another coefficient that may renormalize under RG. Moreover, because these are two different fields, you have to put two different coupling constants here. So these two different gammas, okay? So the gamma is there because the system is out of equilibrium and non-Galilean invariant. Okay.
But I mean, is there a reason why that extra term? I mean that so gamma v is not 1 but something else. So that extra thing is also having the same form as v grad v, meaning that that the renormalization could bring in some other term also. Why exactly this functional form?
Okay. Yeah, absolutely. You are absolutely right. Okay. We started with this, let's say, fairly standard definition of the material derivative. Now, your point is, okay, it's fairly traditional as long as gamma is equal to 1. When you allow gamma different from 1, why there shouldn't be other kind of terms, other combination of velocity, gradient, and velocity? Is that correct?
Yeah,
is that your question?
Yes, yes.
And indeed, so let me tell you, this is a very smart question. And I wish we had asked that ourselves a while ago when we started our calculation. We didn't. So very well done. Excellent question. The RG does generate all sorts of new terms here. Yes. And this is the beauty of RG, that if you forget something, when you do the first coarse graining iteration, you get some new terms. And indeed, these equations are not the full story. I'm not giving you the full story, I'm sorry, but you can check it on the paper. Because when you do the RG on this equation, you generate 4 anomalous terms. 2 are anomalous terms of that are anomalous transport terms, which are exactly all the other possible combination you're mentioning. And other 2 are anomalous terms in the mode coupling part. So let's say different ways of which you can have this G, because your same question could be asked with this G. This G should be 1 if you have standard Hamiltonian dynamics. But here is out of equilibrium, so you put a coefficient G, and then you could ask, well, then you could have other combination of this V cross and so on. And indeed, you do have them thanks to the RG. But it's RG that gives us what are the correct terms now. And fortunately, when you put them back in the theory, the calculation closes. Okay?
Okay.
But yes, so this is very important. So unfortunately, this is not the full story, because when you do the RG on that, you get many other terms, okay? Which I wanted to censor to you, but thanks to this question, now you know that it actually happens. So it is even more horrible than this. So in this theory, you start with 4 fields, well, you remain with only 4 fields, which are velocity and spin, and the hatted terms, because we do Martin C. Jarreau's method, in which you have the hatted term. If you are not fluent with that, never mind. And we have 5 nonlinear couplings, the original ferromagnetic one, the transport couplings, and the inertial spin velocity coupling. But as the gentleman before said, actually the RG then generates even more coupling, but fortunately then the calculation is closed. All these couplings have dimension, scaling dimension 4 minus d, which is the justification to use epsilon expansion. So the good old epsilon expansion, and which is what we do. So an expansion in dimension close to 4, and this is why you say critical phenomenon in 3.99 dimension. At one loop, which is the smallest, which is the easiest kind of approximation you can do here, sorry, is the lowest order of the expansion you can do here, and the only one you can do by using momentum shell, which is what we are using here. We are using momentum shell a la Wilson. You have many Feynman diagrams, and this is why we took more than 3 years to do this calculation. But the good news is that at the end of the day, you do find a new fixed point. And this fixed point has dynamical critical exponent, which is equal to 1.35. All these fixed points now are connected to each other by a complicated flow, but the important point is that now we have a final theoretical determination, which is 1.35, which is down here. And here on the right, you do have the experimental determination. So we were very happy when we got that after more than 3 years of calculation. So remember, these 2 points, probably the red one is the most meaningful one, not because it's closer, but really because LS treats the experimental error in a crazy way. But anyway, this experimental determination comes from doing experiments in the parks of Rome with real cameras, are not simulations. And this is a calculation done with 66 Feynman diagrams. So I think that this agreement between the RG calculation and the experiments is quite remarkable. And remember, no free parameters. We had no free parameters. And this is it's not because we were particularly smart, but this is the beauty of the RG. We didn't fit anything. There is no experimental input. Of course, there was a, let's say, conceptual experimental input, because it was by looking at the correlation function that they had the idea to add inertia in the theory. But we didn't measure anything and then put it into the theoretical equation. There is no fitting free parameters in this agreement. Absolutely zero, which I think is remarkable. Let me go back to universality, because remember, that was the original the question. So does this mean that we have the final critical exponent for all systems? No. First of all, we know this works for different species in this family, okay? But already, if you go to mosquitoes, and we do have data for mosquitoes still unpublished, but Anopheles gambia, these are malaria mosquito. It seems that things are different. It could be different because you are in the lab and not in the wild, but so we still don't know. But for example, we expect certainly this not to be the right critical exponent for birds. Why? Because birds are in a different phase, are in the deeply ordered phase. And we know that we have universality classes that also depend on the phase. So we wouldn't expect the critical exponent of Heisenberg close to TC to be the same as the critical exponent of Heisenberg at low temperature, right? Because you have different symmetries and you have different universality classes. But still is something which, ties together different animals in this case. And if we broaden our horizon, we see that if we summarize all our fixed point of our theory in just one synthetic picture like that, I think it's very nice that we are describing on the same tableau, let's say, very different physical and biological systems. So down here you have standard ferromagnets, what is called model A in dynamical critical phenomena. Up here, if you have inertia, down here you have the case where you have no inertia and you are at equilibrium, no activity, no inertia. If you add inertia but no activity, you go to a class of model which is the one of superfluid helium, Z equal 1.5. On the other hand, if you add activity but no inertia, you get the prediction 1.7, which I showed you before, which might be, there is no experimental evidence up to now, but might be what is important in dry, overdamped active matter, overdamped because there is no inertia. And up here, when you switch on both inertia and activity, you get our new fixed point, which is the relevant one for natural swarm. So I think that being able to describe all this in just one, so different natural systems, from superfluid helium to natural swarm, active matter, in just one slide is one of the incredibly profound features of the renormalization group. We couldn't do that in any other context. So the message, the last message for the young people in the audience, which is the one I really would like to stress, if you want to understand this, so if you're thinking about maybe working in biology or physical biology, really don't forget to study this as well. So consider studying, field theory, good old field theory, even at the equilibrium level, because this is not past. Okay, I think that this is still very relevant to study that. These are the people who work with me on that. These are the 3 PhD students who did the, I have to say, quite amazing calculation. Stefania and Leonardo, who are the one in charge of the experimental data, and the other seniors with me were Irene Giardina and Tomas Guerra. And thank you very much for your attention.
Thanks a lot, Andrea, for this very stimulating talk. So we have time for questions. So people can raise hands or directly ask the question.
Can I ask a question?
Yeah, can I see your face? Can you switch on the camera?
Please. Just 1 second.
Okay, don't worry. If this is problematic, don't worry. Okay, hi.
Yeah, hi. So my question was, can Kramers-Werner like duality can exist between flock and swarms?
Sorry, can you ask that again?
So can there will be a duality between swarm and flock? So like.
Okay, I wouldn't call it a duality, let's say, but yes, we are now using, let's say, the same model, which is this one, okay, the inertial spin model, to describe both flocks and the swarms. What is the difference? The difference that in this model you have a noise. So the noise is like a temperature, if you want. And by changing the amount of noise, you can be in the ordered phase if you have very small noise, and then you are in the flock case. Or if you have larger noise, you are around the ordering transition, which is the swarm phase. So it's not a duality, actually. It's the same model, but studied at 2 different values of the temperature, if you want.
Oh, thank you.
I had 1 question, actually 2 questions. Can I ask? Hi, Andrea. This is Anirban from IIT Bombay. Yeah. So can you just show that phase diagram of your, sorry, not phase diagram. I mean, that RG flow diagram. I just wanted to check about the number of the parameters. It's 3D. So what are the 3 things? I mean, this is in the 3 parameter space, right? Meaning your RG, you are converging to some value, that red spot. And what is on the X, Y, and Z axis?
Okay. Yeah, sorry. So let me comment a little bit more on that. So let's start from this point here. So this is standard ferromagnets, okay? So in standard ferromagnets, you have no activity and no inertial mode coupling, okay? So forget activity for the moment. So now let's add a new axis, which is this axis here. So this axis, which is the inertial mode coupling. So this would be the G, right? Or actually the effective coupling constant connected to G. Okay, I'm not writing it here, which is G over gamma, but anyway, it's G.
Okay.
Yeah. So then you switch on that. And what you have is that you have this new axis, and here you have the fixed point of superfluid helium. Okay, so as you see, the arrow goes from the blue point to the black point. So this means that if you have some friction, anyway, this is unstable against the friction. Okay, if you put some friction. Okay, then if you now here add some activity, and so this is now this axis, activity, then you get out of this plane. So this is the zero activity plane, which is why we call it equilibrium plane. If we now add activity, you flow to the new fixed point, okay? Now,
Okay. Yeah.
this plane here now then has non-zero inertia, non-zero activity, okay? But you see that I put here underdamped plane. So remember that you have a friction, and the friction is what I call eta. This is a reduced friction, which we call eta hat, but never mind. So then, if you had a little bit of friction, asymptotically, you anyway go always back to the overdamped case. So in the infinite time and infinite size case, friction always wins over inertia. So you do expect that all these fixed points, the superfluid helium or quantum antiferromagnets and the swarm fixed points, are unstable against adding some small friction. But what is the point? The point is that these are finite size systems. So when you have a finite size system and you are close to a metastable fixed points, you may have a crossover, meaning that you do not flow away unless the system becomes very, very large. So if you start close enough to underdamped case, you first flow to these fixed points. And if the system is not infinite, that is the critical exponent that you observe. But then if you get a very, very, very large system, you always flow to the underdamped case. So we believe that our fixed point, which is unstable against friction, so remember friction is here, okay, is what dissipated spin, is unstable against friction, as it should be, because remember, it must go to the Witzek model for large friction. But if you are in a finite size systems, you may see the neighbor of that. And the proof of this is exactly in these correlation functions. If you were in the vicinity of the overdamped fixed point, you wouldn't have this kind of correlation function, but you would have an overdamped correlation function. So this is why we think that the correlation function is telling us that we have a very small value of the bare friction. So, and because the system is finite size, we have not been flowing here.
So by friction, it is the friction of the medium.
No,
is that right?
No, I'm sorry. Yes, I'm sorry about that. It's an effective friction acting on the spin. It's not a friction of the of the medium. It's not the real. It's not the real viscosity of air.
Okay.
Exactly like inertia is not the real mass of the.
Okay, I understand.
In our new model, inertia is a generalized inertia. Is the resistance to turn, and this is a highly effective parameter because the way they turn, the actual maneuvering of the animal is very complicated, very different between birds and midges and so on. But it is not the friction. It's something which dissipates the spin, the generator of the rotations.
Okay, thank you. I had one general question, but but not exactly on this talk, but on swirling systems. Like there are some experimental evidence which were taken from drones. You might have seen this fish. Sorry, this what is it called? This the sheep flocks, not exactly sheep herds. So there they see a lot of, I mean, since you were talking about spin, a lot of intrinsic rotation in the herd, especially close to a wall, or if you perturb the system somehow, they seem to rotate. Does that anything to do with these spins that you were talking about? Or rather, can those systems be modeled using the same spin language you were talking about?
I don't know, first of all. I don't know for sure. The same kind of rotational behavior that you describe is present also in some species of birds, like in swifts. Swifts do these huge circling patterns to go back in the simnay, to get into the simnay. I think that yes, the spin could be an ingredient of that. However, I think I remember that there are models of collective behavior even without inertia and spin that can reproduce the rotating patterns. So remember, the rotating pattern would be a rotation really in the external space. So a sort of standard angular momentum, which of course may be connected to the inner angular momentum, but is not the same thing. What we introduce is the internal angular momentum. Again, the two things could be connected, but it's not strictly, I think it's probably not a necessary condition to describe that, because I remember that there are some models, well, typically done by biologists, so with many parameters, but I would say that there are models which can give you what they call the milling phase, even without having the spin structure that we produce. The spin structure that we introduce is really crucial to have a different way to propagate information. And the idea basically is that if you don't have inertia, you could have a change in the direction of motion of 180 degrees in one time step. In the Vic-check model, that is allowed. So if all your neighbors are going in the opposite direction, so imagine that you're flying alone, and now a group of neighbors arrive in this other direction, and say, oh, they're all going in the same direction. I instantaneously change direction. This is what Vic-check does. So this is what it means to have no inertia. What we say is, no, okay, this force is acting on the spin, and the spin is then rotating the velocity. This mechanism is what enhances the propagation of information, which, if you want, is a bit counterintuitive, because you say, if you put inertia, which means being lazy, you propagate information more efficiently. The answer is yes. And if you think about the domino effect, it's very clear why it's yes, because inertia is the resistance to start moving, but it is also the resistance to stop moving once you are moving. Yeah. So this is why when you have inertia, you have you have linear propagation. If you kill inertia completely, you have diffusive propagation. It's much less efficient.
Okay, thank you. Thank you.
Can I ask something? So you were saying that this is like some effective inertia and friction. So I was just wondering, so for these bacterial swarms, for example, which are naively I would think of as overdamped, like low Reynolds number thing, would you expect to see a critical exponent of around 1.7? Or even for these like whatever these actin motor myosin, sort of like this motor protein and those sort of systems, what would you expect?
Well, no, I would not expect that, because even though this inertia is effective and generalized, yes. However, I think it must have a connection also with the actual dynamics, aerodynamics or swim dynamics of the swimmer. It's not an easy connection, but it must have a connection to that, right? So, for example, this inertia of ours would be connected to, imagine a plane or a bird. It would be connected with the aerodynamics of a turning bird, which considers many things, the shape of the bird, the muscle, the skeleton, the physiology, and the medium, the air. And in this whole interaction of that, it gives you this effective inertia. In the case of bacteria, because the real friction, the real damping is so large, probably, I'm not sure, but I would not expect inertia to be relevant there, exactly because even though at an effective level, there is some trickle-down propagation of the actual Reynolds number parameters down to the effective parameter here. Remember, these are effective theories. I'm certainly not doing an ab initio theory from the shape of the bird to the theory. So I can only speculate. But from what you say, I would say that no, in those cases, I probably would expect that to be overdamped.
So you would expect an exponent of closer to 1.7 rather than 1.
Yes, I would expect closer to 1.7. Yes. But I cannot know for sure. But before doing the exponents, I think they should remember the first signature of that is directly in the correlation function, the shape of the correlation function. When you do have underdamped dynamics, you always see it very clearly from the correlation function. And we see it in swarms, and we see, of course, even in flocks. Flocks time correlations, so bird correlation in times, are definitely not exponential. Now, for bacteria, I don't remember now any study of the time correlation function of bacteria, but certainly there are. but I don't expect those to be strongly non-exponential.
Okay.
But again, you can see directly from there. It's a way, if you want to infer, not quantitatively, but qualitatively, the bare value of the friction, of the effective friction.
Okay. 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 Thank you.
I think I'm still muted. Sorry. Are there any more questions? If that is not the case, let us thank Andre once again for
Thank
this
you.
very
Thank you very
beautiful
much for
talk.
inviting me. Thank you.
And yeah, so I think we can then end the session. And so, Mithun, anything to?
No, I think that's all. Thanks a lot. It was a really wonderful talk. Yeah.
Okay, thank you guys for inviting
you.
me, and good luck to everybody.
Yeah.
Thank you very much for a beautiful talk. Thank you.
Thank you.
Okay. Bye-bye.